You wanna know how to replicate some DNA? Get a parent strand and a protein hand, and we'll show you the correct way. I know you know what it is, it's semi-conservative and it happens in the nucleus. I wanna show you how to replicate some DNA, like DNA replication, DNA replication.
Discovered in the early 50s by Watson and Crick, a process in the cell that's deceivingly quick. Know that it's amazing, let me introduce it. It's DNA replications happens in the nucleus. Proteins, the nuclear DNA is all you need. With them and the right tools, the system will proceed. Helicase, primase, ligase, to name a few. DNA polymerase and SSBs too. With the right method and these proteins no fewer, you'll soon be replicating like rats in the sewer. DNA polymerase deserves a lot of credit. Not only does it polymerize, but this enzyme also edits. Lastly, let's not forget the all-important telomerase. With this bound template, it's always in the right place. By grabbing repeat sequences and adding them all, its job is to prevent the strands from getting too small.
You wanna know how to replicate some DNA? Get a parent strand and a protein hand, and we'll show you the correct way. I know you know what it is, it's semi-conservative and it happens in the nucleus. I wanna show you how to replicate some DNA, like DNA replication, DNA replication. So we have a bunch of enzymes all essential to the method, and before anything else, helicase will be requested to separate the parents and just a little upstream to overcome the twisting to polymerase is seen. Single-stranded binding proteins have a crucial job to do, keep the parent strands apart so the rest of the crew can start replicating. Primase is coming up next to place an RNA primer for the enzyme complex. And DNA polymerase is the star of the show. From 5' to 3' nucleotides go. The leading strand is simple and is synthesized quick, but the lagging strand is done in Okazaki fragments. Once the primer is erased and replaced by DNA, the nicks between the fragments will be sealed up by ligase. And once all is done, we have 2 double helix strands, and that's replication. We hope everyone understands. You wanna know how to replicate some DNA? Get a parent strand and a protein hand, and we'll show you the correct way. I know you know what it is, it's semi-conservative and it happens in the nucleus. I wanna show you how to replicate some DNA, like DNA replication, DNA replication, DNA replication, DNA replication, DNA